Hi, I'm Fleur and I do the Human and Molecular Genetics Masters at the University of Sheffield. We're outside Firth Court at the moment and today I'm going to be doing my labs and my placement at the Julia Garnham Centre. I did my undergraduate here at Sheffield. I did it in biology and I originally chose the city because I loved how green it was. I decided to stay on for my postgraduate because my course offers a lot of lab time and also an NHS placement. I also got a postgraduate merit scholarship to help me with my studies. So for the first part of my day, I'm going to head to the labs. So we're in Firth Court now. This is the cell culture room. We have to culture our cells twice a week to keep them healthy, and we've been doing it since February, so we've got quite a lot of experience now. These are the cells we use for all of our experiments. They're called HeLa cells. They were taken from a patient in 1951 and they are a cervical cancer cell line. I'm just about to culture my cells. This is a cell culture hood, which we use to keep everything sterile. So these are the HeLa cells that I was culturing earlier in the lab. We've done fluorescent in situ hybridization on them. So we can see the chromosomes here and these are nuclei. If we change the filter, we can see the telomeres. FISH is a method that is also used diagnostically and is kind of the follow-up after karyotyping, which is what we do in the Julia Garnham Centre. So I've been able to do a lot of different things this year, including fluorescence, PCR, Western blotting, CRISPR with different yeast strains, and cell culturing, which you saw earlier. At this time of the year, I'm doing my research project, so I'm only in the labs, but before Easter, I was also doing lectures. I'd like to go into a career in clinical genetics, hopefully in the NHS. That's one reason that I decided to do this Masters, because it's kind of given me a lot of different skills that I might need to get into a job. So I've just finished in my labs. I'm going to go for some lunch now at the Students' Union, and then in the afternoon I'll be at the Julia Garnham Centre for my placement. So I've just come back from lunch. I'm now at the Julia Garnham Centre where I'll be doing my placement this afternoon. I work with several other students to help reduce the leukaemia diagnostic backlog in the Yorkshire and the Humberside. So at placement we work to pre-karyotype patient cases, and this is generally leukemia, and this benefits the NHS in two ways. Firstly, we can free up staff time to do other things, and secondly, as we're pre-screening lots of different cases, we can occasionally flag an abnormal case to the Children's Hospital and they can then decide to fast track it. The summer before my Masters started, everyone on my course was contacted about doing a placement at the Julia Garnham Centre, so that's how I got involved. But anyone in the School of Biosciences can do a placement here, and that's undergraduate, postgraduate and PhD as well. So this is a training case. It's anonymous and there's no patient data here, and we have permission to use this. Here you can see a metaphase, and this is one cell of the patient's. We can cut all these individual chromosomes out and then we can karyotype them. I have an incomplete karyotype here and I'm just going to finish doing this now. So aside from the skills that I've gained from doing the pre-karyotyping for the NHS, I've also got to take part in a number of other opportunities. We've got to karyotype some stem cells for a research group, and we're now being included in their paper, which is in Science Advances. A small team of us also won a Team Worker Award, and we also do a lot of really nice socials, and it's been a really great opportunity to meet students from other courses. This is a completed karyotype. We have an abnormal chromosome here. If we saw this in a real patient case, we would flag it to an NHS staff member. So I've just finished my placement. I'm going to head home now and work on my dissertation. Thank you for joining me. Bye!